Hello there, everyone. Welcome to Monday, August 16th, 2021. This is a very interesting video. Let's do a little background on what's going on here. Back in 2020, as coronavirus started to take over and we started to close everything, I became more and more interested in a project I started, I think, like a decade ago, or almost a decade ago, which is analyzing molecular biology. Basically, there's a lot of what makes up our bodies and our cells. It's a string of RNA or DNA. In this case, it's RNA when it's the coronavirus. Adenine, cytosine, thymine, guanine, bases of RNA in order that create amino acids. And so as I became more and more interested in how this works and what's going on, I started to grab sequenced versions of different iterations of the coronavirus. There's one from Wuhan right here. There's one from Italy right here back in February of 2020. And here's one from, I believe, Los Angeles or Louisiana. I can't remember which one the LA stands for, just recently. And I have other files that convert information from the National Institutes of Health, NCBI, I believe that's like biometric part of the website, where you can get the actual accession numbers and get the actual strain of the coronavirus as it has been identified fully. When you bring that into this cube, and this will be another video explaining how those other files work because they're a bit more complicated. I just want to focus on some conclusions actually, because I've been working on this intently over the last few days, and to tell you the truth, I'm really excited about what this is doing. This is fascinating, and I hope to express that to you in this video as we go through and expand what some of the capabilities are with what this thing does. So here we go. Basically, you have genes that we've identified on the coronavirus. So there's the Sars COV2 is about 30,000 nucleotides or bases long. You can use this thing called a codon string to pick where the codons start, because amino acids are 3-character bases and they form codons. Or the codon is what tells you, you just click this codon 1 and you get basically all the amino acids are going to show up by gene for each one of these, or all 3 of these actually, all 3 of the versions that I have in the file. And we can add more pretty quickly. You can add any accession strain, any number. We can just add them to this list and bring them in, and then run them into this cube and basically analyze them like this. So what are we looking at, and why is this really, really cool? Well, there's a lot of things hidden in here, and I'll show them to you in a minute. They're hidden right under here, which is different attributes of the amino acid and the order of things. So we're going to get to that in a minute, but let's talk about looking at a broader view of just everything right here. So what does this mean? This is the order of the gene on the string of all of the RNA. Well, the 5' end is the top or the beginning of this. So if you open that up, there's no product associated with it. It's just kind of the beginning. But what you can do here is you can actually say, all right, let's just look at, actually 5' end is kind of boring, so we're not going to look at the 5' end, but we're going to look at something like the spike protein, all right? This is the S is the spike protein. Well, we can just click S here on this gene slicer, just S. And now we're looking at the spike protein for all 3 of the versions of the virus that we have in the file. They have 1,274 codons, or 3,822 full RNA bases in there. When you're at codon 1, you get this 1,274 number of amino acids. So now let's start to open up all this stuff, and I'll show you the capabilities of what you can do here, why it's fascinating. So let's look and say, well, what is going on? We know that something's going on with the transmissibility of the virus more recently, the Delta variant and stuff. I don't know if this is a Delta variant strain right here, but it is recent, and I would suspect that there are Delta variant strains in here. But we're going to find out probably as I look up more information about this one that we have, which is this one right here. MZ712900.1 is just a version number, but this is the main number. You can look that up on the NCBI website, but watch what happens as we open up what's going on in here. And sometimes this will take a while to process, but sometimes it won't, depending on how much we're looking at. So you start to expand everything, and we're going to be able to compare all of the different attributes of the amino acid and make sure that they're the same, and also find out where they're different, and also see what their states are here. So this is where it gets really, really cool, right? So if there's a 1 here, that means that all 3 of these strains had exactly the same amino acid in exactly the same order of the spike protein in their version of their RNA sequence. So that means they all started with this ATG methionine, which is a hydrophobic initiator. Then there are these hydrophobic amino acids, phenylalanine, valine, phenylalanine, and they're all the same all the way through, right? So that's great. We're having uniformity in the sequence of the RNA. Let's find, oh, wait a minute, something happened. Something happened right here. Something happened around the 55th character of the RNA and the spike protein in the USA version is all of a sudden this switched to arginine from what was theroanine. So it went from a polar to a basic is what happened, because there's a 1 right here when there was 1s here before. Okay? And so that's an interesting mutation right there. That's something that is immediately comes apparent when you start scrolling down. So does this have an impact in the spike protein and in the formation? Well, what was theroanine is now arginine. Well, let's take a look at the images that we have, which are right here. These things right here. I try to visualize what these amino acids actually look like and then the structures that they form at the molecular level. Well, the 2 that we're talking about are arginine and theroanine, right? Well, this is arginine, so that's what it turned into, and it was this one. So it basically grew a little bit. Looks like this one's a shorter little wishbone sticking out, whereas this one is a longer curved wishbone sticking out. So in the structure of the spike protein, all of a sudden we have something extending out a little more than it used to in my mind. Now, I don't know if that is accurate because I'm not a molecular biologist, but I do know that adding this guanine in here instead of a cytosine, which is represented here in what the chemical formulas are for these bases, is we're putting a G where there once was a C. You can see that the mass of the G is a little bit bigger at 151 grams per mole or whatever versus 111. It has more density. It's a bigger molecule. So that's what happens when you replace the G with the C in there. Okay? So cool. We found a mutation or a insertion or something, not an insertion, but some type of mutation that has happened right around 55. Well, what else we got? Anything else we got in here? Here's another one. We have valine has turned into isoleucine. Okay, now here they're both hydrophobic, but here's a slight change. Right? I don't know if that's going to have that much of a big difference. We could look at what isoleucine and valine look like. Isoleucine and valine. Is it isoleucine, this one, and valine is where? Here. This looks like it has just a little extra coming off of one of the wishbones compared to this one. That's what that looks like. So here's more extra stuff hanging off the spike. Is that what it turned into? Because it went from valine to isonucleine, right? Let's see. Looks like, yes, it turned into isonuclein. So yeah, here's something else extending out, growing a little bit bigger, I guess, in the spike. What else we got? Down here, we've got an error on the USA version. We have an unidentified nucleotide, so that's why this is probably not working, but that's probably not much to worry about. Then we have a lot down here, same problem. We have some unidentified nucleotides down here. Whenever you see an N, that's a problem. I can try to remove those triplets, but point is what we're doing here is we're doing a really line by line, amino acid by amino acid, nucleotide by nucleotide comparison, or base comparison. Here we have an issue. We have a leucine arginine transition. This went from a hydrophobic to a basic again, right? We had that at the beginning. That's down at 1354, and this is you can also double click and say, like, where is this? This is actually, if you double click, it'll tell you which base it is. It's base 22,878 is that one that we're talking about. So any other things down here? We've got a threonine turning into a lysine. Okay, we have the loss of a cytosine turning into an adenine there, which is interesting because those two don't usually bond, so that's an interesting change in mutation there. And we got something down here, which is some aspartic acid slightly changing from a thymine to a cytosine, it looks, at the end of that one. We've got some down here. We've got a aspartic acid turning into a glycine. That's kind of interesting. That's an acid turning into a hydrophobic. And something here. We've got a hydrophobic turning into a basic. Again, that seems to be a phenomenon of mutation that has been repeated in a few things here. Seeing hydrophobics turn to basics. And what else we got? Anything else? Something here. That looks like a nucleotide sequence missed, so I wouldn't worry about that. Whenever we see that N, don't worry about it. It just means unidentified. And toward the end, we've got a little aspartic acid change, but not much. So the point of all that is that was just a rudimentary review of the spike protein, right? The surface glycoprotein and comparisons from older versions to a newer version. It is quite easy to import new versions, and that's what I'm going to do over the next few days and really get an idea of what kind of changes we've seen in the Delta variant and how they compare to other things in the past, and then we will find out what's going on. So that's the tool. If you're interested in using this, or you want to learn more about it or get involved with this process, I think it's beneficial for everybody for us to really understand what's going on at the molecular level, and then we can try to combat it. We can build tailored vaccines, we can build tailored treatment plans. It all gets down to this base level of analysis, this granular level of what's going on. It's science, and we can get there, so we'll work toward it every day. All right. Good luck, everyone. May all of your biohacking be positive. Good luck.